just a
Okay, well, thank you everyone who has joined us once again for the IWRS webinar series. I will talk very briefly before I hand over to our speakers, Gabor and Marco, who have a really fascinating talk for us today. I am genuinely quite excited to hear about this. With that, I will turn my camera off and hand over to you guys.
All right, good, thank you. Okay, now the camera is off. So yeah, welcome all of you. Thank you for a few people coming here in person. And we are in Göttingen, Germany, Friday afternoon, so that's a nice way to finish. My name is Prof Lövei, and I'm actually based in Denmark. And now came here to the seminar, and I'm an entomologist, working mostly with ground beetles, and then with agroecology. And we had some history in common with Marco. He was based in Denmark for how many years? 5 years, yes, altogether. But now he's at agroecology in Göttingen. So, but let him a few words to say about himself.
Hi,
Marco.
Nice to meet you. Thank you for inviting us. I'm Marco. I'm a postdoc in the Functional Biodiversity and Agroecology group here in Göttingen in Germany. As Gabor mentioned, I was his former master and PhD student many years ago at Aarhus University in Denmark, and after that I moved to different labs in first in Israel and then in Portugal in the Azores. And since a couple of years now, I'm part of this group, and I'm also, like Gabor, working a lot with the carabids and spiders and in general ground active organisms.
Yeah. All right, good. So we're going to split the presentation in two. I start first, and then at a certain point, Marco will take over. And again, also in my name, thank you for the invitation and the possibility to present this material for us. We'd like to touch on these 4 points. First, a little bit about arthropod decline and a survey or a short summary of the entomological methods which are in massive use today. Then a little bit more about this curious phenomenon. What do scientists who aim to protect arthropods, what tools do they use? And you will be surprised about that sort of controversy there. Then a little bit of an ethical consideration. Why does it matter? Why does it matter what methods do we use? And then if we say we are unsatisfied with some of the tools which are in massive use, what else can we do? What are the alternatives? Okay, so the first point is very brief. I assume that most of the people who signed up or who are present here are familiar with this phenomenon about arthropod decline. But I would like to emphasize here that for some unfortunate reason, mostly it's called insect decline. But I think it's very important to widen it and include, and I think it would be much more correct, at least entomologically, if we talk about arthropod decline. But Paul Eggleton published a review about this in 2020, and he claims that the first papers sort of talking about this phenomenon were in 1970, where he managed to find 3 papers. Then more and more, so by about 2010, more than 160 papers deal with this phenomenon of insect or arthropod decline. There are quite a few very heavily cited papers, some notorious papers which were heavily debated. But the first one is from Germany, actually, where Hallman and co-authors compared simply the biomass of mass collected arthropods in 27 German protected areas, and they detected a massive decline in arthropod biomass. It's more than 60%. And if you consider, or if they considered only the main activity period, the midsummer in Germany, then the decline was even steeper. It was more than 80%. So that sent the alarm bells ringing, and there was a lot of subsequent analysis. Then people realized that we don't have many reliable long-term data sets about arthropods and so on and so on. 2019, Sanchez Bayo and Chris Wickhus published a kind of summary tinged with a personal impression, saying that insects and arthropods are in decline. There are several elements in that paper, very heavily cited, very heavily criticized and debated. But I think the overall personal impression is quite convincing. And for example, one point I mentioned here is that there are very few red list arthropods, but about almost half of those which are showing also signs of decline. And of course, you know, the flurry of activities, and 2021, Wagner et al. Wrote a summary, and if I remember well, that was actually a special issue or section in PNAS, trying to summarize the evidence. But basically, they said it's anthropogenic, human activities from habitat destruction to urbanization in various ways, but all of them negatively influence the abundance of arthropods. So this is where we are at the moment. And when you look at routine entomological collecting methods, many of them are non-selective. There are some, of course, you know, like a pheromone-based collection, which could be very specific, rarely species-specific, but fairly so. But many of the routine methods have been developed without any consideration of the numbers and kinds of arthropods and any possible consequences, because probably traditionally there has been, you know, a massive number of arthropods, and it was unimaginable that any sort of collection would cause lasting damage. But if you talk about, you know, you consider the top left, and that is a Malaise trap, and practically relies on the flying insects. If they hit any obstacle, they will strive to fly upwards, and the collection, you know, devices, you know, is in the top. So any flying arthropods, just because of this behavior reaction, will end up in a collecting vessel. And likewise, you know, many arthropods in a sticky trap, that yellow square, which is glue, coated with a glue, will capture anything which either flies or climbs on it without any, if the yellow trap, as the yellow color is attractive, which for many arthropods it is. And likewise, you know, a pitfall trap, any arthropod climbing or, walking on a surface, if it doesn't notice that there is a hole there, will end up in the trap. So really quite a lot of other arthropods. This is just one example from Marco's, one of Marco's students. Master thesis collected more than 30,000 arthropods, aimed at carabids and spiders, but those two groups actually provide less than half of the total catch. So the bycatch can be quite massive. Looking at the methods further, this is a very famous book on ecological methods, specifically designed, not the latest edition, but the earliers were specifically for insects. And they do mention some methods which are not lethal, but they don't make any consideration. They don't mention that those would be maybe more desirable or less damaging than anything else. So in that sense, the book is basically silent. And this article published in 2022, where several entomologists, that was a meeting in the UK, that they set themselves the goal, what are the big challenges facing entomology in the coming decades? And they did not even mention that entomological methods could be modified or maybe less damaging to arthropods. When we look at the particular segment in the literature, it's conservation science, there are two journals, main journals devoted to arthropod conservation. You know that a Journal of Insect Conservation, and the other one is Insect Conservation Diversity. And of course, there are a number of other conservation biology journals which may have invertebrate papers. So we decided to have a look at those for 7 years between 2014 and 2020. And we looked at the published papers, only the primary papers, so reviews and editorials and debates and all that were left out, checking whether the method used for data collection was lethal or non-lethal. And then we also checked if the focus of the study was a single species, a group of species, or many species. And we also separated whether the study was done in a tropical country or region or non-tropical one. So we evaluated these papers. In one of them, the Insect Conservation Diversity, they published 274 papers during this period. And more than half of them used lethal methods. Only about 30% were non-lethal, and another 12% both. For single species, it was more frequent than non-lethal methods were used. More than half, you see nearly 56%. But if more than one species was the focus of the study, then only about 22% used non-lethal methods. And there was also a geographical region. I don't think it's very surprising. Arthropod world in the tropics, where most of the species are, is less well known. So tropical studies routinely use more lethal methods than non-tropical ones. As you see, here is the non-lethal methods. In non-tropical regions is about 1/3, but only about 12% in tropical regions. And more or less, you got the same picture in the other journal that has many more papers, more than 500. But you see that about half of them use lethal methods, about 7% both, and only about 46% non-lethal. And the same thing, single species focus, only about 15% was lethal, more than one species, 62. And the tropical, non-tropical is the same. So non-tropical areas, only about 37% use lethal methods, but up to 75% in the tropical region. And when you look at the other conservation, here is the summary, you see that by different orders, there are some orders where there is more tradition for non-lethal methods. As you see here, dragonflies, Lepidoptera, so butterflies, and grasshoppers. And here the blue color indicates the prevalence of the non-lethal methods. So it's in non-tropical regions, for example, dragonflies and overwhelming is actually non-lethal method study, but not in the tropics. In the tropics, you see the green color dominates, which is the lethal methods. So even if you break it down to certain orders, for some orders like butterflies, more frequently people study them by not killing them, but not in the tropics and not for many others. When you look at the other conservation biology journals, first, there's hardly anything. You see that more than 5,000 articles in these 4 journals, only 246 were on, not on invertebrates, but on arthropods. So this well-known bias for vertebrates is very, very prominent here, but more or less you get the same picture. Yeah, so single species, certain orders, and non-tropical areas have a higher chance that that study will be done by non-lethal methods. Lethal methods dominate, basically. Now, one thing is this summary here, that most of the entomological methods are lethal, and they feature even in articles which are published in journals which are devoted to conservation. And this geographical and taxonomic sort of bias or particularities are there. Now, why does it matter? Yeah, so the question emerges that insects are very numerous, they can multiply very quickly, they can rebound, and so on and so on. Does it really matter? So we would say at the beginning that it does, no matter what you consider. One thing is utilitarian, right? So insects or arthropods are perform several functions which are very important for us. In this place, I don't have to mention that pollination is a very important one, decomposition, biological control, and so on. So if you go from this point of view that insects or arthropods are useful for us, so they have instrumental value. Can also argue that ecologically they are important because they constitute most of the world's biodiversity. They participate or drive many important ecological mechanisms. So that would be really quite a problematic thing for many ecosystems if all of a sudden they would diminish or disappear. By the way, the two pictures are two recently discovered jumping spiders from Australia, and they're really, really beautiful. As you see that it's not only the birds of Australia are very colorful, apparently the jumping spiders are really nice. Now, the traditional argument was, and it goes back to the French philosopher and mathematician René Descartes, the mechanistic approach, because he claimed that animals are simply machines, and by using the mechanistic understanding and physical processes, you can understand the world, and it doesn't matter because these animals not only don't have a soul, they don't have feelings, they don't feel pain. So Descartes, for example, was not a cruel person, but he performed dissections on live dogs without anesthesia. And you say, well, the dog seems like suffering, but not really, because they are machines. They just seem like they are suffering, but they're unable for this because they're not thinking. Although there was, even at those times, minority opinions expressing that this is not permissible, it's not desirable. Sir William Jones was a very famous Orientalist. He spent a lot of his time in British India, in the colonies, and returned. He became a very prominent intellectual in Britain. And he wrote he gave a seminar at the Asiatic Society in 1793, 1 year before his death, where he writes that it's not acceptable that birds are killed just because they are nicely colored, or even, he says, deprive even a butterfly of its natural enjoyments because it has the misfortune to be rare or beautiful. So he condemns the destruction of life even for scientific knowledge. He says, just because it's rare, just because it's precious, is not enough reason to kill them. But by and large, I mean, this remained a minority opinion. Now, when we consider whether animals are ethical subjects, usually three criteria are mentioned. One is that can they feel pain? The other, do they think? Do they show any signs of thinking? And the other thing is, do they have any interests? Now, the first question traditionally was asked, but we don't know. In 1984, in an Australian journal, there was a review. Do the insects have any neurological mechanisms to feel pain? And the conclusion at that time was that no, they don't. So they cannot feel pain. But we mentioned there are a few recent papers, we only mentioned two here, where this perception is changing and is probably no longer correct. So actually last year in 2023, in Royal Society, one of the journals. They revisited that and say insects have the mechanisms, the hardware to feel damage to themselves, which by all definitions could be reflected as pain. They show behavioral evidence, they have molecular evidence for that. And then a year earlier in PLS biology, they say the conclusion is that the evidence is not always very solid. There is very good reason to believe that insects actually, or arthropods, do have the mechanisms to feel pain. So the question for the first, so the answer for the first question, yes, according to present knowledge, I think they do feel pain. And then at this point, I hand over to Marco, who will explain or expand on the other two questions.
Yeah, thank you. Thank you, Rob. Yeah. Hi. Yeah. Another of these arguments come from Kant. This is one of the classical arguments, who believe that we should only respect those animals who are showing signs of intelligence, who are rational beings. And at the time, Kant did not consider all non-human vertebrates to be rational. But nowadays, I think we all agree more or less that all vertebrates at least are rational organisms, and also some invertebrates. This is an example from the European Union. So within the EU, cephalopods have been they recently attained the status of honorary vertebrates due to the recognized intelligence. So they've been raised to different categories just because of this intelligence, and this implies that we should treat them in a similar ways when we, for instance, want to make certain studies on them, or also for food production, for instance. I'm now going to show you some examples from the arthropod world to try to convince you that also arthropods can be highly intelligent. I mean, I'm sure that you already believe that, but just a couple of examples. Consider, for instance, spiders. A spider that has to repair its web. And then the spider web can be damaged in many possible ways. And in order to fix the web, fix unpredictable damage, the spiders needs to basically apply problem solving, because it's very unlikely that the spider is pre-programmed to solve every single types of damage. It's way more likely that the spider has a general understanding, a general idea of how its web should look like, and then it basically takes decision case by case. How should I repair my web in order to have it the way I want? So problem-solving skills. Similar examples, another type of maybe criterion for intelligence comes from honeybees, because honeybees have their own language, and language is one of the criteria for intelligence. Using languages, honeybee can communicate, indicate the position, the location of the food sources within the landscape, the distance to the hive, and even the amount of resources that are available to decide how many workers have to leave the hive to harvest. So I think we can conclude that there are signs that invertebrates can be highly intelligent as well. So now the question is, do they have interest? And when philosophers talk about interest, they basically refer to the ability of whatever life that can be made better or worse. So every organism has its own needs, and based on the realization of these needs, its life can improve, its life can flourish. And of course, every organism has its own interest. I think the interesting point of this angle is that you don't have to be, an organism doesn't have to be rational or able to feel pain in order to have interest. Plants may have interest, for instance. So we can see that according to all these three criteria, arthropods can be considered ethical subjects. And to summarize, there is basically this branch of philosophy that is value-based, that decide or suggest that we should give arthropods moral consideration based on their value, and their value can range from instrumental value, so the benefits that we obtain from them, but also ecological value, so the value of the species in ecosystem, and also the intrinsic value. So we recognize in this organism intrinsic values, and this can be dependent, but also independent of the valuer. This is actually debated among philosophers. I will now briefly mention another perspective, another ethical approach, you can say, which is not based on value, but is based on virtue. Basically, virtues are those guidelines that tell us how we ought to live our life, right? And some of these virtues are, for instance, compassion or respect for life, and they refer to the outcome of our action. So our action can be positive if they help other living beings flourishing, if they don't interfere negative with their life, or can be negative otherwise. We are now switching topic. Now we are going to talk about what are the alternatives for studying arthropods without killing them. So after we discuss why we should do that, what can we do? And this is based on a recent review that we published with Gabor in Annual Review of Entomology. And in this review, we categorize these various techniques I'm going to show you in a moment, and we try to also underline and stress their pros. And I think that the pros of non-lethal methods are pretty straightforward, because from the animal's perspective, the animal is not killed, there is not damage, and also interference with its life activity is minimized. But also for the researcher, there are obvious pros, because there is no moral burden. There is also another aspect which it is recognized, but I think it's very important. We mentioned it also in the review, and should be stressed. I think that non-lethal methods are actually more suitable than lethal ones to work, for instance, in citizen science projects and programs to engage the public. And this is extremely important nowadays, because it can help us fighting the so-called extinction of experience. So this is a phenomenon, a general detachment from nature or from experiences in nature, which is particularly strong in the younger generation. So in the review, we categorize these non-lethal techniques along a continuum that goes from techniques that damage the organism without killing it, until techniques that are real non-lethal but also non-disturbance technique. And among techniques that cause damage, we have, for instance, a mutilation to extract the genetic material for population studies, for instance. Often it's possible to just clip part of the leg or the elytra of an animal to do genetic analysis. Don't need to kill the entire individual. Another example is clipping, that sometimes is used for markings individual to identify them. So it's possible to use clipping and damaging the individual without killing it. And then we have a set of techniques that do not directly harm the individual, but they may actually also increase indirectly the mortality, so they can cause stress or discomfort. For instance, when we use transponders on the elytra of some beetles to track their movement, we may also limit their ability to escape from a predator. They may hinder their movement, and this can be stressful for the animal, may have a fitness cost, essentially. Another example is the fluorescent markers, like in the beetle in the picture. Fluorescent marker, we don't actually often know, but it's reasonable to think that it may make the prey more detectable to potential predators, so it may increase mortality rates. Often there are not a lot of studies trying to check if this is true or not. And finally, we have a set of techniques, and in this presentation, we focus mainly on those that are real non-disturbance methods. So they are non-lethal and also don't cause any potential indirect effect. Some of these can be relatively simple. You cannot see it from there, but I have here a trunk refugia. I'm gonna share it with the audience here. Maybe you can have a look. Basically, this is a very simple device. It's basically a cylinder made of cardboard roll, and then we attach it using strings, either to tree trunks or to branches. And with this, this is actually pretty common in spider studies. Arachnologists use it sometimes to collect overwintering species or spiders that live on branches, on crevices inside the bark of the tree. And it's very cheap, it's very easy to use, and basically the mechanism is that animals enter within this space between the two layers of cardboard. And I use this technique myself. I use it when I was in Israel, in the Arava Valley, in the desert, and I was supposed to be collecting spider authors, monitoring spiders. And I was actually very surprised to see how many groups can be monitoring using trunk refugia. Of course, there are a lot of spiders. I mean, spiders were the dominant group within this refugia, but there were also several other arachnids, over 8 beetle families. We found Lepidoptera larvae, Hemiptera, Hymenoptera, and so on. So my point here is that I think that sometimes the non-lethal methods may be less frequently used, as Gabor showed a few slides before, also because we are not aware of their effectiveness. We are not aware of their potential use, because there are not many studies focusing on testing and calibrating and exploring the potential use of these non-lethal techniques. And many people go for the safe old way of doing things, despite all the ethical problems with that. There are also, of course, many techniques that are especially very popular nowadays because they rely on higher technology. So technological advances nowadays, it's possible to record even small organisms such as arthropods using camera or audio recordings, and of course these are a bit more expensive, but they seem to have a great potential, and it's very likely that it's gonna be improved very quickly in the next years. In fact, I'm not an expert of these methods, but I would recommend you to check the review by Van Klink et al. That was published in 2022, where he gives an overview of these methods. Here, what I want to stress is that artificial intelligence is not exactly a panacea for for the problem. And of course, it's very appealing. There is great potential, but there are also challenges. And so the first obvious challenge is that the species identification is still very problematic, because it's afflicted by the same problem, the same gaps, the same lack of knowledge that have afflicted the traditional taxonomists all the time. So if we are not aware of the species and we find an unknown species passing by our camera, we still cannot identify it reliably. There is also another problem, which is to find reliable and good training data sets for these algorithms in order to work effectively and correctly. There is also a lack at the moment of public reference databases that will be needed for these techniques to work. And another things that emerge by when we read these articles on artificial intelligence is that many authors seems to envision a system that will combine deep learning together with molecular tools, so that it will be possible somehow to identify the organism, skipping this species ID validation problem by combining these techniques together with molecular tools. Unfortunately, it's not yet clear how. So it's desirable, perhaps it's the idea, but nobody knows exactly how to do it so far. And we also have to remember that currently molecular tools are not particularly effective to provide reliable estimates of arthropod abundance. So maybe it can be used for determination, but if you are interested in abundance or density of organisms, it's still a problem. Another very simple and effective nowadays non-lethal way of studying organisms is actually by observing them. And of course, going to the field and doing everything with visual observation, visual transit is a lot of work. But nowadays there are a lot of platforms that allow to record organisms all around the world, and they are also very suitable for citizen science program. Probably the most famous of this platform is iNaturalist. And there are many others. I mean, yeah, I just provide some examples, some links, mainly from North America, but we also have similar website in Europe. And on iNaturalist, just a few days ago, I checked, and there are at the moment over 50 million observations for arthropods, mostly insects, but also spiders. So it's a treasure, basically, because they can provide a lot of information about distribution of species, presence, absence, and so on. Phenology as well. In the review, we also discuss a different way to reduce research cause mortality. It's not exactly a non-lethal, non-destructive technique, but basically we stress the importance also of using already killed insects, because there exists already a lot of material that has been collected. Gabor before mentioned and discussed our common and our abundant is bycatch, and we could simply make a much better use of it. At the moment, there are no coordination of programs or whatever to make use of this bycatch. So normally what happens is that it's typically stored for sometimes a few years, and then it goes to the trash bin. And it's absurd, it's a pity, considering how abundant it is and so on. But there are also other data that could be used. For instance, insects accidentally died along roads, so roadkills. There are some examples of studies using roadkills data for studying also dispersion movement, but also presence, absence of arthropods in certain areas. And probably what is the most obvious is relying on museum collection, because museum collection have a lot of material in them, and sometimes there are many species awaiting to be described and discovered just within museum collection, which is ironic. I think the hope is that now with technology, there seems to be a direction towards digitalization of this collection, which may make them available also to people around the world, and potentially a great source of data. So I would like to make a clarification because it's also always a hot topic. I also had some discussion with some colleagues when the review appeared. I also been in contact on ResearchGate with some colleagues that was highly critical about this idea of promoting non-lethal tools. But I think there was maybe a misunderstanding. We did not say that we should completely abandon from today to tomorrow non-lethal, the traditional techniques, because it's true that the non-lethal alternatives cannot always replace them, especially when we are interested in species identification. It may still be a problem, but wait for a few slides because I want to talk more about this. So I think what we try to promote is that, first of all, researchers should know that they exist, they can be effective, that sometimes an alternative exists for answering their needs, and potentially that they have to be, but they could be tested, used also in combination, and that in general we should move towards that direction. But of course, it's not going to happen in one day. This is the point that I wanted to make so of course, species identification is often a big problem when you don't have the specimen. With arthropods, it's tricky for most groups, maybe butterflies or dragonflies in the temperate areas of fauna that are well known, as we've seen in the data that Cabo was showing. But I think one question is, what is the parameter that we want to measure when we start our research? So we should start asking ourselves which parameters we need in order to answer our questions. So, for instance, if I'm an agroecologist, if I'm establishing a flower field in a landscape, do we expect the change to happen only at the species level, so at the community level, but or do I expect, for instance, changes in the activity of the organism, or for instance, in their behavior or in their fertility? So there may be different parameters that will still indicate that there is maybe a success in the management that I'm doing it, that do not require killing the individuals. So there are many parameters, and species richness is only one of them. Nevertheless, I also wanted to stress that sometimes species richness may be also avoided because for some taxa, not always, but for some taxa, there are studies showing that genus level, subfamily level, or sometimes even family level are a very good surrogate, and they can be used to basically to obtain the same data and the results that you will obtain by using species richness data. Because in some groups, for instance, ecological traits are already well conserved at the family level. These are just two examples. I will. There are references at the end of the presentation, so don't worry, don't take photos. Here there is another example. So before we're talking about other parameters. Sometimes, especially in agroecology, agroecologists or farmers are interested not really in the identity of the species that perform certain functions, but in the function itself and in how to quantify those functions. So, for instance, here we are very interested in monitoring pollination, quantify pollination or biological control. And for the farmer's perspective, if biological control is provided by species A or species B, may not be so relevant. Of course, there are pros and cons, right? And the slide here is showing a set of tools that we call sentinels. We call this the sentinel approach, where the sentinel can be a different thing, can be a plant, can be a dummy caterpillar, can be a seed. And this sentinel is exposed in the field under standardized condition for a set amount of time, and it provides information on the activity of the arthropods. So it's non-lethal. It doesn't provide information about identity, but it provides quantitative data on certain functions. So what we ought to do? So we think we should probably start always by asking ourselves which kind of parameters we need to record in order to answer our research questions. Immediately after, we should ask ourselves if there exists a non-lethal alternatives to the method that we were originally planning to do, and if collection is necessary. If there is no non-lethal alternative, then we should ask ourselves, is there a target technique that will only collect the organism I want to collect, sparing the others, so avoiding wasteful killing? But also, is it possible to avoid mass killing, even of the species that I want to collect? This is particularly important maybe here when we work with agroecology, because we're actually working on beneficial species we would like to preserve. We would like to convince the farmers their importance, and we don't want to eliminate them completely, eradicate them from the landscape. Another question that I think we should always ask, generally is a good practice in science, is what's the minimum sample size that we need? I mean, this is a general ecological, statistical question, but it's also potentially very important to save the life of many organisms. And finally, if after we ask all these questions, we end up that we had to use a little techniques and unfortunately we have bycatch, then we should at least plan how to use this bycatch, how to maximize its use, how to make the most out of it in order to avoid the wasteful killings of organisms that will eventually end up in a trash bin. So to summarize this talk, so we can say that, okay, arthropod decline is happening, and we need to convince the public that even the life of small creatures like arthropods mattered. And if entomologists, if we study insects using non-destructive tools, we think that we have a greater chance to succeed, because we set an example, a positive example to replicate. Moreover, there are several ethical reasons why we should try to foster this transition from lethal to non-lethal methods. And we believe that actually one of the grand challenges for the next decades is to develop, to test, to calibrate the use of these non-lethal tools, to make them more effective, to understand if we are missing some kind of information compared to traditional technique, what are we missing, and so on. And finally, we think also that journals and research funders could actually help this transition if they raise their standards a little bit when we submit a proposal or we submit a paper. As I promised, there are a few references at the end of the talk. So then you can maybe you want to check some of this paper more in detail. And of course, we are welcoming every question or discussion, and we want to thank you very much for inviting us again. Thank you.
Yeah, go back to.
Okay.
Yeah. Well, yeah. Okay. Good.
Yeah, I'm back. Well, thank you so much for that incredible talk. I personally have quite a few questions just on the back of that. I think this is such an important topic, and I'm really, like, personally, yeah, on a personal level, delighted that both of you are publishing about this. We'll start with our audience questions first. So regarding the review about lethal versus non-lethal approaches in arthropod studies, I was wondering at what point of the study were insects mostly killed? Was this during the capture, after the capture, for tissue collection? So yeah, at what point during these studies was lethality introduced, I guess?
During collection.
Yeah, that's the most common one, even before identification. So yeah, many collection methods actually kill the arthropod. Then you go and collect the material, and then you start sorting or doing something else with it. the majority of these methods. Yeah, that's.
Okay, so I guess in terms of reducing lethality in our methods, like preventing collection or collecting like through these non-lethal methods you outlined, you think is the best intervention we can make?
The period, trying to be more selective. When do you need? Where do you need? And as Marco mentioned, what is the necessary sample size that you need to answer your research question?
Yeah. On the back of that, I'll plug the IWRS research guidelines and methods section on our website, which have some guidance on sample size estimation, and also an upcoming seminar that we have on how to conduct sample size estimation using simulations. So put that in your diaries. That'll be publicized soon. Moving on, I'll stop doing my plugs. How do we ensure reproducibility of the results if species that are difficult to identify have no preserved specimens?
Yeah, well, probably that is it's not an absolute thing. So it's not no preserved specimen, and then probably once, for example, the DNA-based identification will become accepted and more sophisticated, it could be many fewer preserved specimens necessary, and at one point it might be sufficient, to provide documentation of, for example, a DNA identification.
And also, when this is connected basically with what I was mentioning before, that we don't think we have to abandon completely. I mean, sometimes there will be situations where maybe you have to collect because you don't have alternative. I think what we have to change is our approach to the research, that we start asking yourself questions that we normally don't really ask. We just start with the traditional technique and without thinking.
Yeah. So similarly, relating, I think, to perhaps the use of lethal methods in some instances, we have a question which is, bycatch has been an invaluable resource for discovery of new species. Should we not focus on A, that bycatch is included in research permits such that it can be legally retained, and B, that bycatch is generally properly preserved in museums or other permanent collections?
Yeah, I mean, yes, I think maybe this question was typed before Marco.
I think so.
We discussed that, and yeah, we completely agree. And I think already it would be a step ahead if the bycatch could be explicitly considered during a project planning. Yeah, so we agree with the comment.
Exactly.
Okay, so next question we have. Great presentation. Are scientific journals making any advancements in terms of controlling for proper use of methods regarding the question asked in the studies and use of bycatch? So again, I think maybe this was...
I mean, they have some guidelines, some of them.
Yeah.
But then when we look at the paper, at the articles and the methods they use, it doesn't seem that reflects very well the guidelines in a way.
But I think we are on the cusp of a change here, because there are some soft guidelines in I think insect conservation diversity. It's at least it gets a mention, but I think pretty soon with some push, these will be easy to put in the instructions to authors. I would think that funders would be a bit tougher nut to, so that the proposals consider these things, but it's maybe a longer process, but it's I just cannot see that it will be completely ignored.
Okay, next one we have. Great talk. Is there any data on the proportion of insects killed by researchers versus killed by pollution and other non-scientific human reasons?
No, I don't think.
I had a discussion with a Dutch carabidologist, and which is, just trying to argue that they were actually running a very extensive pitfall trapping survey of carabids in the Netherlands for about 50 years. And so they preserve the material, they have the numbers there. And he sort of argued and sent me some calculations that actually during going to the field by car, considering X number of carabids killed during the travel, that just going regularly to these places killed more carabids than the pitfall traps. So then, and this could even be credible, but I think the point is not the numbers, it's the attitude. I don't think that we can go very far if we want to convince the normal citizens, saying you really have to be very careful and consider and protect the arthropods in your garden or in your everyday thing. But actually, I'm an entomologist, so I'm exempt. I'm telling you what to do, but I'm not following what I recommend for others. So I think it's not a matter of, it's not a numbers game. It's a valid point, but I think it's an ethical point. The ethical argument is stronger, I consider. So
yeah, I mean, we actually have a couple more questions that are on a very similar theme. So I will. We're running out of time, so I'll refrain from reading them out. Apologies if you type those in. Which is essentially asking this question of how much impact, say, even on a population level, are entomologists having? But I think Marco's sort of mentioned this explicitly in his talk. This convince others of their importance, right? I think we have a responsibility as authorities on this to act in a way which we would like others to act to, and it's kind of incumbent upon us to sort of lead on this, perhaps. Okay, got time for a couple more, I suspect. Thanks for the talk and many compliments. This subject is urging in this time of biodiversity crisis. I would like to point on bioacoustics. I study Orthoptera, and we more and more use bioacoustics as a non-lethal method to assess species diversity. There will be an international workshop on Orthoptera sound recording in the Museum of Verona next July. If you can come, you are welcome. There you go. You have received an invitation to a conference in Verona.
Entomologists are actually some of the few virtuous entomologists who mainly study arthropods without their organisms without killing them. So yeah, we'll see. But thank you so much.
Okay, and next question. Thank you very much for the presentation. I could ask a superficial question, but I was wondering, for what types of research fields is it still necessary to use lethal methods applied even to a very large sample to study arthropods? Who is most in need of applying these techniques?
A taxonomist that I have to describe species need the sample, but one category maybe.
Yeah. As things are today, I mean, an estimate is that we know, let's say, less than 25% of biodiversity on Earth. Most of those are arthropods. And at the moment, this description of new species cannot be done in a valid way, because the tradition is that description has to be based on a physical specimen. So probably description of biodiversity at the moment cannot sidestep, or not yet. In vertebrates, it's slowly changing, because I think, for example, a new bird species doesn't have to be collected. I'm not 100% sure, but I think that is now a photograph or documentation is sufficient.
I don't know, but if I can add something to what Gabor says, and I'm sorry that I don't remember the author, but I think it's Princeton University Press that published a book of a British naturalist of 1800 that they call it iconotypes, and some of the species were described and given the status of good species based on the drawings, watercolors. But it's very exceptional. Maybe butterflies are, easier than other groups.
Yes.
Check it out. It's I can maybe of the book, but yeah, iconotypes. If you type it in, I'm sure you will find it because there are not so many books like that.
No.
Okay, well, I think with that we are just about time, so we will release everybody to enjoy what is left of their Friday afternoon, or the rest of their Friday if they're in another time zone. I'm being a little Eurocentric there. I'd like to thank Gabor and Marco very, very much for that excellent talk, and invite everyone to join us again next month, where we will be joined by Jeff Sebo, who I don't think we have a topic title yet, but it will be some kind of consciousness, philosophy, animal ethics type angle, I think. So yeah, thank you all very much for joining, and see you very soon.
Thank you for inviting us, and good weekend, everyone. Thank you,
All right.
Craig. See you.
Bye now.
Bye.
Bye bye.